Good afternoon, everyone. The topic of today's lecture is mode of DNA replication. I'm Dr. Helyanthus Verma, as working as an assistant professor in Department of Zoology, Ramjas College, University of Delhi. I welcome you all in the lecture series. Before moving on to the understanding the mode of DNA replication, I want to bring your kind attention towards the role of Rosalin Franklin in DNA discovery. As we all must know that Watson and Crick published the model of DNA in 1953 and received the Nobel Prize in 1962 for the DNA structure discovery. But the name behind that was Rosalind Franklin, who was an English chemist who produced beautiful X-ray images of DNA at King's College London. Her 51st photograph was the one who helped the scientists to discover the DNA structure. The photo was acquired around 100 hours of X-ray exposure from a machine Franklin herself had refined. Franklin was really good in X-ray crystallography. But what happened then? If she produced that image, why she did not receive that credit for that? That happened because she had a personality conflict with her colleague called Maurice Wilkins, who in January 1953 disclosed her 51st photograph without her permission with competing scientist James Watson, who was working on his own DNA model with Francis Crick at Cambridge. She did not complain about this at all, likely the result of her upbringing. Franklin, after that, she left the King's College in March 1953 and relocated to Birkbeck College, where she started working on tobacco mosaic virus. In the next 5 years, she published around 17 papers on viruses, and her group led to the foundation of structural virology. She died very early at the age of 37 due to ovarian cancer, might be because of the so long X-ray exposure while doing X-ray crystallography. Her work will always be admired. Before moving on, I want to discuss a brief about the DNA structure, which we all must be knowing that the DNA is basically made up of the sugar phosphate backbone along with the nitrogenous bases, which are of four types: adenine, thymine, guanine, cytosine. Adenine and guanine are called as purines, whereas thymine and cytosines and uracil, which is there in the RNA structure or RNA, these are basically the pyrimidines. Adenine pairs with thymine with the two hydrogen bonds, whereas cytosine pairs with guanine with the three hydrogen bonds. You can see how beautifully this structure has been designed or has been there, which you can see that the nitrogenous bases are attached to the sugar molecule at the first position of the sugar molecule, whereas the phosphate is basically leading to the chain called as 5' to 3', in which the 3' oxygen of a sugar molecule is attached with the 5' phosphate molecule of other sugar. So sugar, phosphate, sugar, phosphate, sugar, phosphate, it basically forming a backbone of the DNA structure. That DNA structure are basically the double helical structures, and which is producing the major and minor groove, and the base pairs are present at the center. One more thing I want to tell you that it runs, as I said, that it is making a bond with 5' phosphate to the 3' region of the oxygen. So basically it is 5' to 3', running like 5' to 3', and the other strand is running like 5' to 3'. So basically they are anti-parallel in nature. Now, what would be the mode of replication? Basically, three different models have been given in this regard. We can just see. The first one is the semi-conservative replication, in which we can see that. So basically, I'm just giving you the model, and then we will define. Just think about it. We will define that what it was the exact or the correct model of DNA replication. What is the correct model of DNA replication? We'll get to know. So basically in the first model, which is called a semi-conservative replication, the parental DNA molecules, which you can just see, get separated. Both of the DNA get separated, and each of them developed one strand of the new daughter DNA. Okay, you can further see that it is further developing the second generation of progeny, it is further developing, and then it again we have one older and a one newer. So that is basically the semi-conservative model, in which a parental molecule gets separating and then they are producing, they are leading to the replication of the other molecule or the newer molecule. So we'll be having a DNA molecule having one old and one new strand. Here we can see the second model, the conservative replication, in which we just focus on the parental DNA molecule, which is just the same. Now in the conservative model, it has been defined that the parental DNA molecule will remain exactly the way they were, whereas similar DNA molecule will be created as a daughter molecule. So the daughter molecule will be entirely the newer one, whereas the parental molecule will be entirely the older one. So in this way, the next generations, the molecules will be remaining their entire position or entire, you can say that they will remain as they were before. So the newer molecules, both of the helical structures are getting produced, but the older one remains the same. That is the conservative replication. The third method which was given is dispersive replication, in which basically what was defined at that time, that it must be the breakage of, there must be a breakage of parental DNA molecules into small pieces, and those small pieces are basically leading to the regeneration of the leftover species. So basically what is happening, that these are breaking into the different parts, and then these different parts are creating the producing the leftover parts and rejoining and producing in double helical structure. You can see that it has produced these two structures and then further produced these four different structures. All are dispersive in a way that they have a mosaic pattern of having the older DNA molecule and the newer DNA molecule. So these three replicative models were given after the DNA structure was defined, because the next step was to identify how the DNA is replicating in the organisms. But the answer for this, that what exactly the mode of DNA replication, was given by Meselson and Stahl. Meselson and Stahl, in their experiment on DNA, for understanding the DNA replication, what they have done in 1957 at California Institute of Technology, they basically used the 15N isotope of nitrogen and 14N isotope of nitrogen while growing the bacteria in a media. I hope you must be knowing that the bacteria are supposed, while culturing the bacteria, it is supposed to be grown on a particular media from where they get the nutrients for the growth. So in that nutrients, if you replace some kind of the molecule or some kind of the nutrient, then that of course will be incorporated into the bacteria. So what that particular thinking, with that particular thinking and the knowledge, what they have done, they first grown the bacteria only on 15N ammonium chloride, which was given as a sole source of nitrogen. So what they have done, they have grown the bacteria only with 15N, and that for a longer time, so that the entire DNA will get the only heavy isotope. Okay, so they have grown it for a longer time. After that has been done, after they achieve in which the DNA must be only of 15N, what they have done, they washed the bacteria and incubated the fresh medium with light, 14N light and 14N containing compounds. So what they have done, they now replace the medium, and now the fresh medium is having 14N as the sole nitrogen source. Okay, so they also took out the samples in each of the generation and then find out that what would be the mode of DNA. It must be a little confusing by now, but how they have found out by using this, but it will be clear in just few seconds, in few minutes, not seconds. So afterwards, did DNA was extracted and subjected to equilibrium density gradient centrifugation. Okay, so they extracted the DNA and did the DNA density gradient centrifugation, and in this procedure they basically mix the DNA with cesium chloride and centrifuge the double stranded DNA molecule according to their density. Now basically, as we talked about the three different models, semi-conservative, conservative and dispersive, they might have got this particular kind of the probable result they thought of. Okay, so they basically planned that what would be the result, and then compare this with the result they get after the experiment. So you can just see the semi-conservative model, what would have been the result. We can just see that in the first generation, if the model is semi-conservative, because I should talk about the parental first, that in the parental one, of course, because the organism has been grown for the only with the heavy DNA, then the band must have formed at the place where it was supposed to have of for 15N DNA. Okay. Now in the next generations, or in the generation as far as the semi-conservative model is concerned, the first generation must be having a hybrid kind of the DNA, right? Hybrid kind of the DNA. Why? Because if you just remember the previous slide in which I just said that this is the parental molecule and this is basically the hybrid kind of the DNA. So why hybrid? Because it is having one strand of 15N and one strand of 14N. You can just consider this particular slide or this particular picture as black one as a 15N, whereas the red one as the 14N. So it will be more clearer to you that how this is leading to the formation of the bands in density centrifugation. Here you can see, again I'm coming to this, that first generation is giving you a hybrid band, whereas the second generation is basically giving you a light band and a hybrid band. So both of the intensity of both the bands are same, right? In the third generation, you can just see that the lighter band intensity is increasing. Of course, it is increasing because there will be more and more of the lighter bands in the successive generations if semi-conservative is concerned. Now in the third generation, in the conservative model, what I have to talk to you about it, that in the conservative model, the person will be getting only 15N and 14N bands, not the hybrid one. Why so? Because if you remember that there will be only two DNA molecules, two kind of the DNA molecule, entirely 15N or entirely 14N. That's why the two different bonds is corresponding to these ones. Next to this, there will be more of the 14N DNA in the next successive generations, because 14N double-stranded DNA will be more and more in the generations. Now let's go to the third one. Let's go to the third one, in which we are having a dispersive model. We are just comparing the dispersive model over here. That in the dispersive model, the first generation will be similar to the generation of the semi-conservative. Why I'm saying this? Because it will be having a similar kind of the pattern as the hybrid pattern semi-conservative has. I'm just focusing here that it is having a hybrid kind of the DNA in the other way, because half of the portion belonging to the parental DNA molecule, whereas half belongs to the first generation or the daughter cells. So that's why the hybrid molecule which has been formed will give the band over here only. It is similar to the semi-conservative. But in the next successive generation, you will find a great difference. Why? Because in the second generation, there will be more of the 14N, but it will remain attached with the 15N also. So that's why it will not form the clear band at 14N position. So the band will be shifted a bit towards the 14N. In the next generation, of course, there will be more shifting towards the 14N and not towards any other side. Whereas so you can clearly compare and define this dispersive model with the conservative and the conservative with the semi-conservative, and vice versa. So I hope it is very much clear that how the different banding patterns might have been obtained if it belongs to the semi-conservative, if it belongs to the conservative, if it belongs to the dispersive. Okay. So, but what exactly that they got is the most important question over here. They basically got the semi-conservative kind of the pattern, in which in the first generation they got hybrid, in the next generation they got hybrid and light, and the third generation they got hybrid and more of the light, and in the next successive generation the lighter band, the density of the lighter band increased. So that's why it was very much clear to them that the model of DNA replication is basically the semi-conservative. The similar experiments were later also performed on the eukaryotic cells to identify that whether they are also having this kind of the banding pattern or not, whether they are also having semi-conservative mode of replication or not, so as to confirm in case of eukaryotes, and the similar pattern was found. So that similarity we also share from the bacteria. we share many of the similarities as far as the DNA replication, DNA transcription basically, and the protein production is concerned. So there are multiple things, but with today's class I just wanted to focus on the modes of DNA replication, how to identify how they found, and what is the route, how they find the right mode of replication, and how we found out that what is the right mode of replication. With this, I want to thank you all for listening and stay tuned.